So whatever we run on the gel and transfer them to the membrane, there are a single strand. That's the purpose, because now at the next step, we want to add a complementary sequence. So they're going to bind to the RNA and DNA we are trying to detect. Then we can visualize them by, if either it's the fluorescent or the radioactive, we will be able to visualize them. The important note here is there might be tons of RNA and DNA there. Some of them, they don't bind to the membrane. We don't want them. We do series of washes. We get rid of them. And also, now we have some of them, they are not containing the sequence that we are looking for. The complementary sequence we already designed, which is called probe, those probe containing this exact sequence and exact nucleotide that we are looking for. So these probe will attach to those RNA or DNA on the membrane that they have the complementary sequences. The rest that they don't have the sequence that we are looking for, it will be eliminated. And we won't, by eliminating means that we won't be able to see them. At the end, what we see on the membrane, it's only those one that the complementary strand will be able to hybridize to our RNA or DNA of interest, and we will be able to see them.
Thank you very much. We have another question as well. The question is, why two original strands of DNA will not rejoin together in polymerase chain reaction?
Okay, that's a very good question. Let me bring this one. So when we have in the polymerase chain reaction or PCR, what is happening when they denatured? So there are a very long strand of DNA, and they need a specific. The previous question, if you remember, it was how we determine the annealing temperature. So the temperature we need to these two long strands to anneal together again, that's why we pick an annealing temperature for the primers, that the primer only will anneal. And remember, these primers are very short fragments that will be able, and then actually they can conquer this. It's kind of a game. It's kind of a race. They can conquer, and then because they are very small, they will be able to attach to the single strand DNA, and then start the rest of the process. But for annealing and then attaching to those two long DNA strand, we need a different temperature that it's not provided in the reaction, so they won't be able. And it may happen some other area of the DNA that we are not interested in, they may attach. But again, in the next cycle, when it's repeated again, when we heat them, because remember these three steps, 95 degrees, annealing temperature, which is 55 to 62 or 65, and extension 72 degrees, these gonna repeat it over and over. Even if there is any chance that these can anneal, it's again at 95 degrees, again they will be separated, and then right away the 55 degree to 65 degree will cause the primer to attach and then amplify that piece of DNA or the fragment. So it won't be enough time or in the ideal temperature for the two strands to just anneal and then go back, anneal together.
Thank you very much. Now we'll be taking a very last question for this session. And the question is from Pritha Das. She is our student. Her question is the next generation sequencing useful for transcriptome analysis? If yes, how can it be done?
Next generation sequencing, yes. The answer is yes. It's useful for that purpose, but it's not really the area of my expertise. So I know that this is being done, but how it's being done and how the experiment is being done and the analysis being done, unfortunately, I won't be able to answer you that. But yes, the answer is yes.
Thank you very much, madam. With this, we have come to the end of the first lecture. Thank you again, madam, on behalf of organizing committee for a valuable talk. Thank you very much.
You're very welcome. It was my pleasure to be here. Have a good evening. Bye.
Now we'll be moving forward with the second lecture of the session. For that, I would like to welcome Dr. Pallav Sengupta. He will be speaking on COVID-19 and its effect on male reproductive system. Dr. Sengupta is a senior lecturer in physiology and head of services research in Department of Physiology, School of Medicine, Mahasa University, Malaysia. He has published more than 120 research articles and several book chapters with current Scopus H index of 23. He has been listed in the world's top 2 scientists by the Stanford University ranking 2020. He has been ranked second among infertility research experts in Malaysia by ExpertScape 2020. He has also been awarded the highest research publications award by Mahasa University, Malaysia in 2019. He has obtained several research grants, including the Fundamental Research Grant Scheme, Ministry of Education, Malaysia. We have a great news as far as today that Dr. Sengupta has been awarded with another highest research publications award by Mahasa University today itself. Congratulations on adding another feathers in your cap, sir. So with this, I request Dr. Sengupta to begin with his lecture. Over to you, sir.
Okay, thank you, Dr. Pasang. Thank you for inviting me, and thank you for your kind introduction. Today, what I'm going to discuss, that is on the COVID-19 and its impact on male reproductive system. So let me share my screen. Okay, so I believe you can see my screen. As mentioned earlier, that I'm going to discuss on COVID-19 and its impact on male reproductive system. Why I have chosen this particular topic? Because on this particular topic, we have reported several research works. We have proposed several hypotheses. And my research interest, that is basically reproductive medicine, and the topic on the COVID-19, this is, I consider, very timely. And as I mentioned, that we have already done some research, and I'm very fortunate that still we are conducting some of the research with our collaborators in the #1 lab in andrology lab in the world, that is American Center of Reproductive Medicine in Cleveland Clinic, US. So in the student's perspective, I would like to say maybe they can take this one as their connector for endocrinology or maybe reproductive physiology in some of their aspects, some of their knowledges. And they will also come to know that how a virus, how this very novel virus is impacting on male reproductive functions. So as I told that we have already published several of the hypotheses. One of our hypotheses I will discuss today that is already been selected as most popular clinical sciences review 2020 by reproductive sciences. So however, I will try to keep it a very basic for the understanding of the students. So SARS-CoV-2 and COVID-19. By now, we all know that what is SARS-CoV-2 and what is COVID-19, when it has started, and how it is impacting the overall population, the global population, our Indian population, and so on. So this is not the first time that when we are getting exposed to SARS-CoV, that is severe acute respiratory syndrome coronaviruses. We have already experienced other coronaviruses, their impacts on our global population. In 2003, we had first got exposed to SARS-CoV. Then in 2012, it was MERS-CoV, which actually impacted severely on the population. So what are these coronaviruses, these SARS-CoVs, basically? These are beta coronaviruses, and whenever they impact on a population, they try to spread from one species to another, and continuously they try to mutate themselves. So in that way, they change their infectivity, and in case of this novel coronavirus also, we can see that with its increased infectivity, it's continuously mutating and its fatality is continuously increasing. So the first case of this particular coronavirus infection, SARS-CoV-2, was detected in December 2019 in the Wuhan city, China. And since then, it's like it's spreading like forest fire, and since last 2 years, it has killed millions of people. And considering that, on 11th March 2020, World Health Organization has declared it as a pandemic. So we need to know what is SARS-CoV, what is these coronaviruses. These are basically the RNA viruses. And the RNA is covered by an envelope and a nucleocapsid. And these coronaviruses, they have spike proteins, which are actually the sites which bind with the host cell receptors. And with that, it actually invades to the cell and causes all the damages. So there are several coronaviruses that can impact our human physiology and definitely our reproductive system as well. So regarding this particular disease, COVID-19, coronavirus disease 2019, it is generally caused by SARS-CoV-2. And like other coronaviruses, it is also covered by spike proteins, as I told, and which contains variable receptor binding domains. And these RBDs, they bind with the ACE2 receptor, that is angiotensin converting enzyme 2 receptor, in different cells. We express this particular receptor in different organs, different systems, including our heart, lung, and so many. And if the expression of this particular receptor is more in any of the tissues or any of the organs, that particular cell, that particular organ becomes more susceptible to this viral infection. Regarding its origin, it has been found that it is generally mutated. It is very much closely related to one of the viruses that is called RATG13, which is evolved from bats. So it can be assumed that it is, it came from bat, but there are so many hypotheses are coming out nowadays, which are, showing some other ways. So, but regarding its origin, it can be assumed as it is mutating from this particular virus that it is. And other than humans, some other animals like ferrets and pangolins, they are also susceptible to this viral infection, which actually pushes us to think that maybe this one of these animals could be the intermediate host. Could be. There is several hypotheses are coming out, so we cannot say it with 100% that it's from the ferrets or pangolins, but as these animals are susceptible to the SARS-CoV-2, it can be assumed maybe they can also serve as the intermediate. So this is about the origin and spread regarding this particular virus. And now when somebody gets infected by this virus, how this particular virus comes inside the cell. So as I told that this virus, SARS-CoV-2, it is covered by, and coronaviruses, they generally get covered by spike proteins. And the spike proteins attach with ACE receptors, ACE2 receptors. And this ACE2 receptor actually gets cleaved by another protease that is called TMPRSS, serine protease, which is actually which cleaves the ACE2 and makes the way of the virus and the viral material, basically the viral material, to the cell. And interestingly, the expression of this particular serine protease is androgen dependent. And this gives us the clue that could be this particular cause could be the reason why the males are more susceptible to SARS-CoV-2 infection, more susceptible to COVID-19 than females. And till that, it has been found that ACE2 receptor is also expressed in several male reproductive cells, in different germ cells, in Sertoli cell, in Leydig cells, which also makes the male reproductive tract more susceptible to SARS-CoV-2 infection. So these are the two basic concepts which actually provoked us to think that maybe these are the causes due to which males become more susceptible to COVID-19 than females. This is about the general occurrence and their spreading. So this particular diagram was published in Lancet by Lee et al. In 2020. The diagram is showing two aspects. One is the mild infection and another one is the severe infection. What happens during mild infection and how it is actually washed out, and how the severe infection impacts upon the different organ systems, and how it ultimately causes the systemic disruption. So in case of mild infection, when the viral load is very less, that time it has been found that when it comes in through the respiratory system, the resident macrophages in the alveoli, they are strong enough, they are potent enough to fight against the virus, and they can evoke both innate and adaptive system, immune system, to kill the bacteria, the virus, to destroy the virus, and also to wash it out. So if the viral load is less, there is no further need of other immunological cells to come in and to create any immune response. Whereas when the viral load is higher, that time what happens when it comes in through the respiratory tract, when it comes to the alveoli, the epithelium, endothelium border of the alveoli and the capillaries, it actually becomes disrupted. And when it becomes disrupted, the fluid of the blood, it starts to exude it towards the alveoli, and the viral material, it also gets disseminated in the systemic circulation. So when the systemic circulation receives this viral material, what it does, it actually starts secreting some of the pro-inflammatory cytokines and chemokines, which attract father cells, monocytes, and macrophages. And what happens again when these cells come, they further secrete the cytokines and all these, and it it results in a cytokine storm. Nowadays, I think we all know, we have come to know, everybody as a layman as well, the term of cytokine storm that found in a patient who is infected with SARS-CoV-2. So this cytokine, it then causes a systemic inflammation, resulting in a systemic a septic condition, and affecting the different other vital organs as well, different other organs as well. So there is a distinct difference between the mild infection and the severe infection. In the severe infection, all our organs may get affected through this systemic inflammation. So this is the way how the virus, when it is coming in through the respiratory system, it goes to our system, systemic circulation, and affecting all our other systems. So now we came to know that definitely it's SARS-CoV-2. It's going to affect our respiratory system. The name itself says it is severe acute respiratory syndrome creating virus. It will definitely affect our respiratory system. And with the mechanism, we came to know that yes, it is affecting other systems as well. But what about the gender variation? As I told before, 2 slides back, that the ACE2 expression in different cells, it actually makes that difference. If the cell is it's expressing more ACE2, that will become more susceptible to SARS-CoV-2 infection. And if and as I also told that the serum protease TMPRSS2, that particular transporter, that particular enzyme, its expression is also androgen dependent. So with those concepts, as I mentioned before, we started to think that all the researchers, they started to think that maybe males are more prone to this infection than females. And the record of its occurrence in male to female, it shows that the ratio is about 2.7 to 1. So males are more prone to this. And regarding fatality, yes, male are more prone to the fatal effects of this virus. It ranges from 1.6 to 2.8. And there are several evidences, several studies till now, and evidence-based literatures, different meta-analysis and all those. They have established that there is a connection between SARS-CoV-2 and male infertility. Later today, we will discuss that how it impacts on the different aspects of male reproduction. So studies, the earlier studies, what it showed that this virus causes injury to the seminiferous epithelium, seminiferous tissue. It causes the Leydig cell degeneration. It's very important because Leydig cell is the major source of testosterone, we know. And if the virus is degenerating Leydig cells, it may cause hypogonadism and other consequences. That may affect spermatogenesis and so other factors. So in few cases only, so to establish all these things, we need to find out that is the virus is going inside the testis? Is it residing in the semen? So some studies only have reported till today that yes, it goes in the testis and it resides in the semen. But most of the studies, I repeat, that they are giving negative report that that this virus is not found, its RNA is not found in testis. But there are ways, if it is not coming inside also, inside the testis, there are ways in which, through which it can affect male reproduction. So in some of the diseased patients, it has been found that it causes orchitis. In 2003 also with SARS-CoV-2 infection, it was reported that SARS-CoV causes orchitis. And in this particular case, in 2019, 20, and 21, in testicular autopsies of the deceased patients, it has been found that the fibrin microthrombi are present in the testis. So this could be due to the viral load, because these are from the deceased patients, and definitely their viral load is huge. And when the viral load is very high, there is a probability of the virus and the viral material to come inside the testis and other organs. So, however, overall, whatever the organ the virus is coming in or no, it is impacting. It is impacting the male reproductive functions, and that's why all these structural changes, functional changes are found. Seminiferous epithelial damage, Leydig cell count decreasing. All these are found in either in recovering patients, in deceased patients. Later we will discuss. So. In testis and epididymis, in the autopsy testis and epididymis from the deceased patients, it has been found that it causes epididymitis, an inflammatory reaction in the epididymis, and testicular interstitial edema, which is very similar in a way that I discussed for the respiratory system, because it disrupts the barriers. When the barrier disruption is there, definitely the viral material can come in. It is also found in the testis that exudation of the erythrocyte has happened. Okay, and this is about the deceased patient, we can understand. But the thing is, in a recovering patient who has recovered from the COVID-19, Lee et al. In their publication in 2020 has reported 39.1% of the recovering patient had reported with oligospermia, decreased sperm cell count. So oligospermia has reported in 39.1% and 60.9%. This figure is very high. This 60.9% of the recovering patients presented with higher number of seminal leukocytes. Till that, I have not found any report that it is causing leukocytospermia or not, but this particular information indicates that when the testis is getting more leukocytes, definitely there is a probability of generating free radicals and creating oxidative stress. So that is another way how this particular virus can damage testis, can damage the cells, the germ cells and other cells inside the testis. So we have several evidences that it is causing, but till that, again, I'm mentioning that the viral dynamics inside the testis, inside the seminal plasma, is not yet detected. The disease patients, they have higher viral load, so that people may argue that maybe due to that it's happening. But why in recovering patients? So the viral dynamics till that for male reproduction, for testicular functions is not yet revealed. So we will wait for that how it actually comes, or how the wash of the virus happens in testis. So regarding the effect of this particular virus or this particular disease, COVID-19, on semen parameters, I have tried to list down some of the studies. These are showing the studies published in 2020. And as I mentioned before, that one study in 2020 had reported that it has been found in the semen samples by Lee et al. And in 2021, it has again, the RNA of the virus has detected in the patients. Okay, so there are two reports which we have recorded that is describing its presence, either the virus or the viral material RNA in the semen of the recovering or the deceased patients. In this particular case, it is a recovering patient for Gassi et al. And for Lee et al., it was deceased patients. So this particular event we have already published in one of our recent articles that is describing on the probability and how the SARS-CoV-2 can contaminate the semen in different ways, not only in the patient, also the persons who are working in the andrology lab, who are also getting exposed in doing different experiments with the seminal plasma, with the semen of the infected or recovering patients. So these are some reports which are indicating, as I told, very few studies are reporting its presence in the semen, while all others are reporting negatively to its regarding its presence in the semen or testis. So now, if whether it is coming to the testis or it is coming to the seminal plasma, that is another question. That is the direct way how it can impact on the reproductive functions, the fertility in men. But the occurrence, the pathophysiology that I have described at the beginning that comes through the reproductive system to other organs, that actually gives us the way that it can impact on male reproductive system in several other ways. Here we have proposed one hypothesis that we have already published last year, that when the virus comes inside the inside any cell, what it causes, it actually causes the inflammation. It causes the release of several cytokines, and this inflammation can indirectly affect different male reproductive organs. It can affect epididymis, resulting in orchitis. And orchitis, again, indirectly can cause the generation of free radicals, oxidative stress, and all. The infection can also cause the psychological stress in the patients, in the infected patients. And the psychological stress can again generate the oxidative stress. And several antiviral drugs, till that it has been reported, like ribavirin, they are also causing oxidative stress. So if they are, all these components, all these infection-mediated or inflammation-mediated changes, either in the testes or in the accessory glands, or the other comorbidities, or the treatment-induced oxidative stress is generating inside the male reproductive system, it can definitely disrupt the functions of sperm. It can definitely destroy the material, genetic material inside the sperm, can cause DNA fragmentation of sperm. It can damage Leydig cells that may reduce the testosterone production. It may damage Leydig cell to impact on the spermatogenesis. So this particular mechanism we had hypothesized based on the published information, which is showing the SARS-CoV-2 mediated oxidative stress. And that oxidative stress, that pathway, oxidant-sensitive pathway, is generally by the NF-kappa beta pathway, by binding with the toll-like receptor type 4. So which causes further cytokine release and the cytokine storm, inflammation, and all these. So maybe for male reproductive system, we do not have till that the exact original report that it is causing oxidative stress, but for other systems we have the report that how the virus is generating oxidative stress and how that oxidative stress is impacting on. So if we put both of this together, we can come up with this particular hypothesis. This one we had proposed in 2020, and it has published in this year. And I was showing at the beginning, this particular report has been selected as the most popular clinical sciences review 2020 by Reproductive Sciences Springer Nature. And this will be displayed during their meeting in July 6 to 9. So the next aspect how this oxidative stress or all these changes can affect sperm DNA fragmentation. The sperm DNA fragmentation is one of the parameters which can affect male fertility. Other than the typical, the classical semen parameters, sperm DNA fragmentation is one of the major contributors which can result in male infertility. So unfortunately, till that we do not have any original report because we can understand that in recovering patients, it is quite difficult to do, because in those patients, maybe the viral washout have already happened, and for the diseased patient, the recording is a bit difficult. So we will wait for that. However, the mechanism what we had proposed and we had published last year in Contraception and Reproductive Healthcare, that proposing is this inflammation. When it is causing the cytokine storm, it may result in oxidative stress. Or as I showed before, epididymis can get affected, like SARS-CoV infection in 2003, which can result in orchitis. Orchitis in turn can generate oxidative stress, and several other comorbidities as well. So all these contributing factors, when they contribute to the generation of free radical, definitely it can result in sperm DNA fragmentation. And definitely when the sperm DNA will be fragmented, the fertility potential in that recovered patient or the infected patient definitely will be lowered. So now this is another concept that relates. My first discussion, what I was telling that that has already proposed, and we have report on that TMPRSS, this particular enzyme, its expression is androgen dependent. And this diagram, it is showing, this is also prepared by us and published this year in Human Fertility. It shows that the expression of TMPRSS is via the androgen receptors. When the androgen level goes high, that time it gets the benefit of it and its expression increases in cells. And whenever its expression increases, definitely now we can relate the story that when the expression of the TMPRSS is increasing, ACE2 expression is already high in our reproductive cells for males, it will follow the pathway that I had showed before. And that may result in the disruption of male reproductive functions. Now, the other way that is relating reproductive physiology with endocrinology is that several reports have showed that the viral amino acids, they mimic ACTH. Because we are discussing on stress, psychological stress, oxidative stress, what we can now think of. We can think that maybe when the stress will be higher, our cortisol level will be higher, one of the glucocorticoids. So is it working in that way? Is the HPG, hypothalamo-pituitary gonadal axis, is cross-talking with the hypothalamo-pituitary adrenal axis? And in that way, this disruption is happening? So to find out, we had searched for the reports that had showed that the viral proteins, viral amino acids, they mimic ACTH. And when they bind with the ACTH receptor, definitely ACTH will not be able to bind with its receptors. And as a result, all the adrenocorticoids, their level will go low, including glucocorticoids. So including glucocorticoid, cortisol, their levels will go low. That means this virus is causing stress, psychological stress, oxidative stress, but it is not allowing the system to cope up with that particular stress. It is decreasing cortisol. It is not allowing that cortisol to come into the play and can prevent the system, try to prevent the system from this damage. So what happens when the glucocorticoid level goes low? That time, there is a connection between HPA, HPG. Our students may later go through that if somebody is suffering from oxidative stress or other psychological stress, what happens? This cortisol, increased cortisol, decreases LH level, gonadotropin level, that specific gonadotropin LH level. But as the ACTH is not able to bind with its receptors, the cortisol level remains low, and it cannot decrease the LH level. So LH level remains very high. I forgot to mention about one of the reports that came in 2020 by Ma et al. That is showing that the LH level, the testosterone LH ratio is decreasing. If I'm not mistaken, in 81 subjects or something like that. So in those patients, they have reported testosterone level is going low, LH level is going high, FSH level is going high. So that time, if you try to correlate that, yes, testosterone is going low, but if the LH is going high, then why the testosterone is coming to its lower levels? So how the testosterone level is decreasing? So in that way, and by following all the mechanisms, those are published already in different ways, in different reports, I'm sorry. It has reported that this particular virus can directly impact on the Leydig cell count, as I mentioned before. So if it is decreasing Leydig cell count, it is directly damaging the testosterone producing cells, and definitely the testosterone level will go low, direct effect. So it is not through the HPG-HPA crosstalk, because the hormone, this particular virus is blocking that particular way of cortisol mediated decrease in Leydig of LH secretion, and when the LH level goes down, testosterone will go down. In that way, it is not playing its game. So its game is through its direct effect. So the thing is, another way is also there, that is when some cell is fully utilized by the virus or the viral material, all the ACE2, surface ACE2 are fully utilized by the virus. What happens? That time, the backward conversion of the angiotensin to angiotensin, it never happens. And this angiotensin 1 or angiotensin 127, that was actually in, it was helping in steroidogenesis and spermatogenesis, that again has got disrupted. So when the angiotensin 2 receptor is fully utilized in maybe in testis or Leydig cell or any other cell, what happens? Angiotensin production becomes lower, and when the angiotensin 1 or 127, its concentration becomes lower, it cannot help in the testosterone production through steroidogenesis. In that way also, testosterone level is going down. So this particular mechanism is describing that it is not working through the normal HPG-HPA crosstalk. And this one we have published in this year in Human Fertility. And this is showing that how the testosterone level may decrease in either recovering or in the infected patients as well. And that is following the same way and supporting the findings of Ma et al. In 2020. Ma et al., they had reported decreased testosterone level, increased LH level, a lower testosterone to LH ratio, and all these. Our hypothesis is supporting it. So we will look for the other reports that will describe the actual way, how it actually happens, or will report the same things, how the hormonal levels are decreasing without the help of this HPG-HPA crosstalk. So the same thing that we had proposed in another publication that is denying this crosstalk last year. In that case, we have not taken the detailed aspect of the hypogonadism or the androgen receptor mediated activation of TMPRSS. Those things we had not discussed in this publication, but we again proposed that it is not through the testicular adrenal crosstalk, and it has also again got accepted. So these are the various ways through which the SARS-CoV-2, the virus or the disease caused by it, COVID-19, can impact male reproductive system. It can decrease the Leydig cell population. It can decrease the Sertoli cell function. It can disrupt the hormonal balance, can cause hypogonadism. It can cause sperm DNA fragmentation. It can alter the semen parameters. In the table I showed that the RNA of the virus or the virus was not directly found in the semen. But if you look carefully, you will find all the findings are reporting that after the infection, semen parameters are declining. So there should be some way that all the mechanisms what we have discussed, either together or independently, they are working on the male reproductive system and causing and impacting on male reproductive functions. So the take-home message from this particular discussion is that the emerging evidences are we are finding that several original reports are coming in, and they are supporting our hypothesis or the mechanisms those we have already published. And there are several ways through which the male reproductive functions can be disrupted. And we have discussed already that the ways through which the testicular inflammation can cause the disintegration of the seminiferous epithelium, can cause the inflammation in epididymis, can increase the apoptosis. Yes, apoptosis, I have not mentioned that, yes, ACE2, if its utilization is more, when the angiotensin level is lowered, that can also decrease, can increase the apoptosis of the sperm cells and can reduce the sperm viability. So there are different ways through which the virus can affect and may cause the subfertility or infertility in men. Till that, we have not got any report of infertility, but for subfertility, we have enough evidences, and emerging reports, they are showing that how it is impacting on. Okay, so these are the references that I had used, including our published reports. And thank you so much. Thank you, everybody, for listening carefully.
Thank you, Dr. Sen Gupta. In this time of pandemic, we are being hit by wave after wave of COVID. We are all very much interested in learning the effect of this COVID-19 on human system. Dr. Sen Gupta, with his lucid explanation, solved a lot of our queries. Thank you again, Dr. Sen Gupta, on behalf of the organizing committee. Now we shall begin with the interactive session. We have a couple of questions coming from the audience. As such, the first question we have is from the Dr. Upasana Das Adhikari. She's our next speaker in a line. Therefore, I request Dr. Das Adhikari to directly address your question to Dr. Sen Gupta. Will you please, madam?
Yeah, sure. Hi, Pallav.
Hi, Upasana.
Nice to meet you. My question is, like, you have mentioned this Lee et al., if I remember correctly, the 39.1% patients with this. Do you know, like, where is the cohort? Like, which country did it represent? And, like, is it a multi-cohort or is it like a monocentric cohort?
I think
what is the... Sorry, go ahead.
The study was conducted in China, and the cohort is... I exactly do not remember that how many subjects were there, and for the infected or for the deceased or for the controls. But as I remember, maybe total was around 50, and only 6 patients were out of 50, could be 34 or something, they responded, and 6 subjects diseased, and in those 6, they have done all these autopsies and everything.
Okay, I did not read the paper, but you might have known, like, do they mention any comorbidities of these patients or like any kind of other diseases which could have affected in the cohort? Like, without knowing the cohort, do you think like you should believe in this number? Because it's quite a high percentage, right? To
not
6 out of
be ignored. Yes. Regarding the comorbidities, I exactly do not remember that if the deceased patients were suffering from it. But some other reports are there which are reporting on the comorbidities. Regarding the Lee et al., exactly, I do not remember about those 6 patients because without knowing that, I cannot say that maybe there is. So there are several reports which are indicating, may not be in malreproduction, that comorbidities are connecting with the oxidative stress generation.
Okay, thanks. Yeah.
Thank you very much, sir. Again, we have another question coming from our colleague itself. What she says is that in both male and female, oxidative stress and other side effects are there. As ovum or follicle is farther away from blood than sperm, they are less susceptible to damage. That is the question. Are they less susceptible to damage because of being more far away from the blood?
There are several actually causes that can happen because, again, I would like to repeat that there are very limited studies based on we can actually answer that yes, it is happening. The answer that we are giving that based on either published reports or some are assumptions, those are correlating with its effects on other systems. So regarding ovum, the first thing is its ACE2 expression, which is quite lower than testicular expression of ACE2. And regarding the expression of ACE2 in testis, we know that testis is it's a population that is a cellular population that is mixed with germ cells, Leydig cells, and Sertoli cell, and so on. So there are individual expressions and the reported higher expressions specifically in Sertoli cells, germ cells, and Leydig cells, they make it more prone. Second one is for males, I'd like to answer first. The testosterone level is dropping, and that could not protect the male reproductive cells from this damage. So all these things are happening in male. For females, there are reports which are showing the expression of the ACE2 is lower than males in ovum. And regarding the production of the estrogen, which is, we all know that it's a potent antioxidant, and that may protect the either the ovarian cells or maybe other supporting cells which are controlling the female reproductive function. Regarding females, the most of the reports, they are related to pregnancy and its passage from the mother to the fetus. In non-pregnant women, its effect, how it is prevented through estrogen-mediated way or through decreased SCE2 expression or some other ways, it is not yet reported. So we can only assume maybe estrogen protecting more and lower SCE2 concentration. And also the androgen level is lower, which is not helping in that way as it is helping in males in TMPRSS expression. So the viral invasion is not getting that benefit, and that is generally found in males also. So maybe these are the ways through which the SARS-CoV-2, the female reproductive system, is protecting itself from the SARS-CoV-2 mediated damages.
Thank you, sir. We have another question with us that is by Rachana Sharma. What she says is that there is a blood testis barrier. So what is the intensity of the infection as such?
There was no report regarding the Sertoli cell till now. Very limited reports were describing about the effect of the virus on Sertoli cells. But recently there is a report came out, I think in this month itself, that is analyzing all the probabilities And it is showing that it is causing the Sertoli cell number decrease, and it is damaging the blood-testis barrier. And it is reporting that it can affect spermatogenesis in that way. So this is the original report. This is the evidence that we have already. But if we try to assume it by the available hypothesis in different systems, that also supports it. Because the thing is, when the viral material comes in, that time, as I told, that it tries to damage the epithelium. And when the seminiferous epithelium is damaged, that is reported in several studies that I had showed you, definitely there is a probability of the damage of the blood-testis barrier. So when the seminiferous epithelium is getting damaged, degenerated, the barrier will be disrupted. And that can make the way of the viral material or the virus to come inside the Sertoli cell, and the other consequences we can understand.
Thank you very much, sir. We have another question from our student, Kinja Sarkar. What she says is that since you talked about the TMPRSS2 activation is dependent upon androgen, therefore, can androgen deprivation therapy be anyhow helpful?
The thing is, whenever you are depriving the system from androgen, you may stop the viral infection up to a certain extent, but you are depriving the cells which are dependent on androgen. And you can save the patient from the SARS-CoV-2 infection, but you can damage the cells, the cellular function of male gonads. So to protect the subject, to protect the patient from the SARS-CoV-2 mediated infection, if you think that we can make the patients fertile or infertile, that could be taken as. But the thing is, several that kind of treatments are coming in, which are indirectly affecting other systems. So we should be very, very careful while we are giving any kind of treatment which is either affecting the receptor or the hormones or something. So in that way, I do not think that it can help the male patients who are recovering. We can help them to survive, but their reproductive functions, I don't think we can help them.
Thank you, sir. Now we are going to take the last question for this session. It is from Dr. Sutapa Chakraborty. What she says is that it is being asserted that the COVID-19 impact is higher in male reproductive system than in female. Yet, as a general participant, I want to know, can we authentically generalize this as the COVID-19 has its impact on human, both male and female reproductive system, though not necessarily causing infertility?
Okay, so there are two things actually in this question. One is the occurrence and the fatality of this particular disease in males and females in general. And second one is reproductive effects, both males and females. So regarding the occurrence, the prevalence of this particular disease in males and females, yes, we have evidence that is not related to reproduction, but we have evidence. I have already showed you the data that how the occurrence is more if I'm not mistaken, it's about 2.7 to 1 regarding its occurrence, and fatality is 1.6 to 2.8, like that. So we have data that yes, male are more prone to it. But regarding the reproductive system, again, I'd like to repeat that regarding female reproductive system, there are not much published reports. Based on that, we can conclude anything. Whatever we can assume or anything that will remain hypothesis. And as I answered in one of my previous questions, that could be due to the decreased SEE expression, or if the androgen level is lowered in females, the TMPRSS activation will not happen, and that will not cause the SCE2 activation that way, or maybe estrogen-mediated protection. That could be the ways through which females are more protected. But again, these all are assumptions. These are not, these are hypotheses, what we are discussing. We have to wait for the reports to come out, which is describing about its direct impact, because it is more prevalent in men, less in females, so how the females are protected and how their reproductive system is protected. We have to wait for the original reports.
Thank you very much, sir. Thank you again, sir, for your valuable and elaborate discussion. Now we'll be moving forward with the next lecture. Thank you very much, sir, once again.
Okay, thank you so much.
We are being overwhelmed by the number of questions that is being put forward by the audience, but since we are running short of time, therefore we cannot address all the questions. We are very much sorry from the organizing committee side. So moving forward, we have come to the last lecture of the day. This is the final lecture, and this lecture will be delivered by Dr. Upasana Das Adhikari. Dr. Upasana is presently postdoctoral fellow at Howard Medical School, USA. She has completed her PhD from Hebrew University of Jerusalem, Israel. She has been awarded with the prestigious Syngene Fellow from Max Delbruck Center of Molecular Medicine, Berlin. Her research interests include gut barrier disruptions during HIV pathogenesis, immunometabolism, profiling of tissue-resident T-cell. Dr. Das Adhikari will be delivering her lecture on host microbiota interaction modeling of the gastrointestinal tract. I request Dr. Das Adhikari to begin with her lecture. Over to you, ma'am.
Thank you for the introduction. Okay,
Welcome,
I'll start sharing. Just let me know if I am visible and audible.
You are visible and audible as well.
Okay, cool. Okay, can you all see my full screen?
Yes, madam.
Okay, cool. Without any further ado, I won't waste more time. We're already, I guess, running behind the time. Okay, so good evening and good morning for myself and whoever is in US. So today's talk will be on host microbiota interaction modeling. And since all of you or most of you are students of physiology, rather human physiology, I want to take you to a little different angle to see how our physiology works. So human physiology means you're studying all the organs, as we all know how it functions in physiological condition like homeostasis and also in pathological conditions. But the thing which we are getting to understand, and I guess that most of you are already aware now, that we are not living alone. We do have more bacteria living in our body. And this bacteria, what I'm talking about, are commensals, meaning that they symbiotically live with our cells. So if you see the number of bacteria and number of human cells, we are only 10% of those microbes, microbiota. So basically, we are less in number to them. But then the question is, how are we even living with them? Because if you see, from the previous talk, that with this pandemic, how a tiny virus can disrupt our whole life, or millions of lives across the globe. So that is a pretending question, how then we are alive with all the huge number of microbiota. So this is a human being depicting different organs where we see how bacteria. So today I will be talking only or mentioning only bacteria, but it's not only bacteria. We do also have as commensals fungi and also some viruses, retroviruses living with our body. So not going to the pathogenic parts, but these are all commensals. Remember, these are all living symbiotically with ourselves. So there are so many organs where the microbial load can be distributed. And from here, I will only take an example for the GI tract. This is just because my interest and also the interest of the time. And also, why I think like this is an attractive site of studying, because my talk will be focusing on the modeling of a disease. So since we heard like the first talk from Sarah, who explained about the molecular techniques, I will be showing you different molecular techniques which we are using and also are being used to study and understand this modeling. And also like from the previous talk, as you heard from Dr. Pavlov, how he explained about what are the hypotheses. So I will be giving you little and bits by bits examples of how this pandemic is looking and how the virus has infected us with different models. So to study and to begin any disease or any pathological condition, one most important thing is you need to have a model system to study, because you cannot do experiment on human beings, right? Ethically wrong. Okay, so coming back, merging everything, today's attractive site and the point of note is the gastrointestinal tract. So what is the GI tract? I think nothing to introduce you. Most of you are the students of physiology. So you know what is the GI tract. It is one of the biggest organ, not one, like quite big organ, from the oral cavity to the esophagus to stomach and to the small intestine and large intestine and rectum, etc. It has like a huge part. So going deep into the larger intestine, how it is morphologically made up of is a structure like this. So that's how inside the GI tract, the cells are oriented and they are structured as. And if you see here, what I wrote here is the epithelium, is you have been hearing from the previous talks also. So epithelium is one of the tissue types, or it's one of the cellular types, which is usually covering most of our organs, or most of our solid organs are made up of epithelial tissue, and which is consisting of stem cells. So again, stem cells means the cells which proliferate by themselves and which can build the whole organ from scratch. Okay, so why is this organ of interest, or why is it necessary to model this organ? So GI tract is not only one of the biggest organ and has so many functions in our body, but it is also one of the organ which has the highest microbial load. Since I started the talking, like this talk about the discussion about how the microbes exist without human cells. So being this microbial load high in the GI tract, this is the perfect model where we can study this interaction. And another important thing is that this is also the organ which sees the food, like something from the foreign or from outside, every day in its life. So we eat food and we ingest it and it goes inside the GI tract and it's like moving around with the cells of our host along with the bacteria. So that makes it like an unique site to study, particularly this host interaction or host microbe interaction. Okay, so of course, a site or a tissue can be interest to any one of us, but how would one try to understand that what is the perfect model or perfect disease what you're going to study? So since I am from India, and of course it is like we're the largest economic country ruling in this world, the most disease or the tropical countries face is a burden of GI and liver diseases. And one of the most burdening thing and concerning thing is child diarrheal conditions. Like we hardly care about diarrhea because it's like a day-to-day daily consumption thing for us, for ourselves. But actually when you see from very top notch, this is like one of the triggering and one of the concerning conditions, and that can actually kill. And it is one of the highest killing also in children, not only in India, but across like various countries. So this is like from 2017 per capita statistics, which show that which countries have the highest load of this gastrointestinal or intestinal diseases, and specifically like in the children per capita. So if you see where India lies is this one. I'm not sure if you can see my cursor. Let me just put the... Okay, so yeah, this is India where we are lying, and this is our death rate with the diarrheal condition. So that is one case. So we are concerned, and that is also important why we can study that as a model and why we have to think over and again that what we need to do to prevent all this thing. Now, as like a previous speaker also mentioned, we are like a taken by with COVID-19. And yes, it is still an ongoing pandemic in many countries across the world. So some countries are doing better with the vaccines and some are still waiting to get vaccines. So again, like what I have studied, okay, so part of my talk will be about this pandemic, and part of this will be going back to the previous pandemic, which is also a concerning thing. So with this present pandemic, which I'm showing here, is that we also see gastrointestinal complications with COVID-19. So these are some of the reported and published data from my last year. And we see that even when we have the GI symptoms, like, for example, transaminitis, ileus, and also other like diarrhea, they are the patients which are more affected with COVID-19. And if you see here in this Kaplan-Meier, which is a survival curve showing that people who have detectable SARS-CoV-2 in their stool were also like dying faster or over the time. And this is done from a patient of a cohort of like 60 patients in a hospital of Massachusetts General Hospital. It's a monocentric study, but there are also reports from different other hospitals across the globe showing this, that how GI manifestations and GI implications are involved with the SARS-CoV-2 or this pandemic. So this is again to iterate the fact or highlight the fact that how actually GI is involved over the years in so many various diseases. No matter whatever disease comes, like there will be one way or the other, the GI tract is affected. And this is to another show that how we are sure and how we are confirmed that the hypothesis, or actually what we are seeing, that the presence of SARS-CoV-2 in the stool is actually killing people. So as you've all remembered, we talked about ACE2 and TMPRSS2. So ACE2 is very well known to be expressed in most epithelial cells, and GI is again not any different. It also expresses ACE2. So here in this picture in the right, you can see ACE2 with the green. So this, if you remember what Sarah explained about, one of the molecular technology is about how would you understand or how would you detect a protein marker. So you can do either Western blot, or you can do immunofluorescence staining, immunohistochemistry, and fluorescence in situ hybridization. So in this right panel, both top and below are one of those examples showing that how SARS-CoV-2 has like infected the tissue. And this is a GI tissue representation of like 2 or 3 patients. And there are also more patients, but we can only represent like few patients here. And thus we prove that actually the virus can infect and actively replicate in the human tissue of colon or gastrointestinal tract. So probably we now understand that how this virus is kind of affecting the COVID, or how COVID is implicating our life. But this is a very new virus, and like we are studying, but we are still like kind of 10% in this study. So to understand more of a modeling system, now I'm going back to a previous pandemic, which is HIV. I mean, all of us know what is HIV. It's an old disease. Many of us think that it is like maybe an extinct disease, but actually it is not. There are still many people living on the earth who are suffering still with HIV. And if not HIV, their HIV is controlled, but they are suffering from other comorbidities, and one of them is actually a GI comorbidity relating to a lot of GI damage. And this endoscopy, this is a representation of an upper endoscopy of one subject, which is showing that how the GI is affected. So we don't have to go in details. It just looks gross, so we can just skip the slide. Another important thing, if you remember, that I started my talk talking about the microbiota. So we know that we are living with the microbiota. So what happens to this microbiota in different disease conditions? And in one such disease, like for example, in this HIV, we have already seen that there is a different gut microbiota composition between HIV positive and HIV negative patients. So if you see the blue, this is representing the microbiota or the microbial population from HIV negative subjects. And if you see the red, this is from the HIV positive subjects. And these are like not one patient. This has been done over like more than 1,000 patients, and also has been published. Like there are tons of reports showing how gut microbiota is perturbed, like their ecological niche is shifted and their microbial composition is changed due to this HIV pathogenesis. So we know that there is some kind of disturbances in them. So there is one that either this disturbance can cause problem to our cells, or either it is our cells which has led or resulted in this kind of disruption of the microbiota. But that is like either chicken or egg. We just don't know yet what is the answer. So we'll keep seeing what is going on in the GI tract. So HIV, which is again was thought to be only a disease of the immune cells. Of course, it infects immune cells. Let me brief you about the immune cells. So the immune cells, or in the immune system in our body, is the system which kinds of helps in our protection. So it is very different than the epithelial cells, which is covering most part of the organs. So HIV is a virus which infects specifically one of the CD4 T cells, which is our immune system. But though it infects directly T cells or immune cells, it does have an effect in the GI barrier. Not only GI barrier, it also does have effect in lungs and also in the female reproductive tract and also in the male reproductive tract. I'm not going into the details of that, but basically the barrier is almost the same. So all the mucosal tissue in our body, maybe it is the vaginal epithelium or it's the pineal epithelium, which is in the testis or in the male reproductive organ, or if it's in the lungs and also in the GI, it all acts the same. There is a barrier of the epithelium. Across one side is our host cells, which is like rest of other human cells. And on the other side, it's the microbiota. So what do we see here? How do we even know that this is disrupted? So we have seen in patient samples, like when we look for soluble CD4, this is one of the marker of, you can just look through in the textbook, you will see what is this marker. This is a marker of systemic inflammation. This is also highlighted in this pandemic like COVID-19, that people who have higher expression of soluble CD4, we know that they are ending up with this term cytokine storm, or the systemic inflammation is upregulated. There is also this marker called IFAB-B. So IFAB-B is a specific marker which you find in the blood only when your GI is disrupted. So from 3 different patient cohort, here we see from US, Botswana, and Uganda, which are like mostly my patient cohorts across the world, we see that the GI is definitely disrupted, and there is also a systemic inflammation going on in these patients. However, if we also see from patients that though there is a GI barrier disruption, but the cell-to-cell junction, which is called the tight junction, is actually intact in HIV patients. And this is a picture from electron microscopy. This is, again, another type of microscopy where you can see the most detailed structure and the morphology of a cell. And we see that the cell structure and its tight junctions are intact. So that raises the more question than what is actually damaged in the GI barrier, which can cause this kind of systemic inflammation. So when we see more patients in detail, and again, coming back to one of the immunohistochemistry or immunofluorescence technique, by staining for cleaved caspase 3, which is here CC3, which is a marker of apoptosis. I know you have been all hearing about apoptosis and all these things and epithelium. So apoptosis is programmed cell death. Most cells in our body undergo a programmed cell death condition, and that is called apoptosis. And we use a marker called cleaved caspase 3, which is classically used across the globe for representation of apoptosis. And here we see that in the epithelial cells, when we compared versus HIV negative patients to HIV positive patients in their colon mucosa, that there is a more number of apoptotic cell deaths. And we also quantify among different patients, and we indeed see that it is highly upregulated or increased in HIV positive patients. So definitely we know that there is some kind of damage or dead cells, which we see in the epithelium of the GI. So we know that not only the GI the barrier is damaged, but the kind or the phenotype of the damage is probably, or plausible cause is increased apoptotic cell death. So we don't have to see the whole details, because my talk today will be making you understand why it is important to study a model. Because again, we cannot work on human patients. If you're studying human disease, you need animal models. Of course, we have mouse, rat, monkeys. We have so many mouse and animal models. However, there are certain diseases like HIV. It is a human disease. It doesn't infect mouse cells. It doesn't infect monkey cells. It doesn't infect rat cells. So how would you study We would never understand this disease. Also, if you come back to the SARS-CoV-2, SARS-CoV-2 is a very specific virus which only infects human. It doesn't even infect rat or mouse or any rodents. So you have to either modify the virus to study this, or if you want to study the unique virus and its infection and pathogenesis, you need some kind of human model. So this is a very, very new or hot things going on in the world, and we have also accomplished this kind of thing. It is something called organoids. So organoids are tiny microscopic 3D organ models. So what today I'm showing you is a mini model of colon or a gut or GI tract, because we started as our interest as a GI tract, but you can pick any epithelial tissue or solid organs and make into organoids. Now, of course, there's tons of protocols for figuring out how organoids are made, and you can find that in, you can Google that or find in some textbooks, and if not, happy to help you or talk about this later. But basically, this is one of the coolest technique which people are utilizing to see how we can study the whole system or the whole tissue in a petri dish underneath the microscope. So this is how we start from. So this is like one of the crypts bottom, and this is how it actually looks when we get or isolate crypts from human samples or human tissues. This is like an enlarged version of the crypts, and this is how after several days it turns itself like in a 3D ball, and this is like in an enlarged view. I can show you a better version. So this is like organoids made from HIV negative and also from HIV positive subject. The green shows F-actin, which is just marking the plasma membrane. DAPI is marking the nucleus. You know, every cell is nucleated except the RBC. And this is a pure epithelial model, so there is no RBC, no immune cells, nothing else. And Ki67, again, it's a classical marker for cell proliferation, just to show that these are live, and I'm not showing you some dead structures. And just to show as a live structure, this is how exactly the organoids look. So now, while it keeps playing, I will explain you that when we study our physiology of the GI tract, we know that GI tract is like a hollow pipe, right? And what is there in the hollow pipe? Food, processed food, microbiota, everything is in that lumen, which makes into formation of stool, and that is excreted. So in this organoids, if you see the hollow in between that green line, is that the lumen. So you can imagine like it mimics exactly the same as that of the food pipe rather than just in a plate. And if one is interested in studying more details, you can also introduce many other factors to this model system, and you can study that. So what we see from this two different organoids, or comparing from two different patient types of organoids, is that in the right hand, their actin cytoskeleton is kind of disorganized. You can see from the green ones, and you should acknowledge that how it is not normal like the one which is in the left. So this shows that there is definitely some kind of disorganization in the cellular structure, and also we already have seen that there is large amount of apoptotic cell death going on in HIV positive patients, and thus we can hypothesize that probably this is due to the apoptotic cell death, but we are not sure. We need to confirm that if it's indeed the apoptotic cell which is causing this kind of damage. So how do we understand if it's a epithelial cell death or there is a damage which we have seen in vivo? Because in vivo, what we have shown before is the patient tissue or pinches, which we do endoscopy or colonoscopy, and we get tiny pinches and we see that. But we cannot work on that every day or always, because once you fix a tissue, it's gone. That's it. So you need to understand the mechanism, and here is the organoid where you can model it and we can study. So if you compare again two different organoids, and we see the cell death by the staining of tunnel. So tunnel marks pan cell death, not only apoptosis, but fixed. So basically